Prime editing, a modern gene modification method. Because of the error-prone method of cutting of both DNA strands used in gene editing, although being innovative, it has been a subject of controversy. All of that is set to change with prime editing. This procedure makes use of an innovative enzyme called a prime editor, which can modify the genetic code in precise ways without severing the DNA, resulting in more accuracy and fewer mutations. With the use of gene editing, researchers can precisely alter the genetic makeup of living things. Many uses for this technology exist, including the treatment of genetic illnesses, the creation of novel medications, and increased crop yields. The present approaches, including CRISPR-Cas9, depend on cutting both strands of DNA, which might result in mistakes and unwanted changes. A novel technique called prime editing seeks to get beyond these restrictions. It may also alter a larger variety of things, like adding or removing substantial DNA stretches. Prime editing, developed by Harvard University researchers under the direction of chemist and biologist Dr. David Liu, promises to be the surgeon that gene editing requires by just cutting one strand when necessary. Early iterations of this method contained restrictions, such as the ability to alter just particular cell types. Prime editing guide RNAs, which act as the cutting tool in twin prime editing, launched an enhanced variant in 2021 that can edit longer DNA sequences, more than 5,000 base pairs, which are the rungs of the DNA ladder. Meanwhile, by pinpointing the molecular mechanisms that hinder primary editing, researchers at the Broad Institute discovered strategies to increase its efficacy. According to the study, the new methods may more successfully and with fewer unintended consequences fix mutations that lead to type 2 diabetes, Alzheimer's, heart disease, sickle cell disease, and prion disorders. Prime editing has a more dependable DNA substitution, insertion, and deletion method, which allows it to fix more complicated mutations. A crucial development is the technology's capacity to operate on bigger genes, which account for 14% of all mutation types. Despite all of its potential, Dr. Liu and his colleagues realized that the technology is still in its infancy. To employ the technique for medicines in the future, they are still performing more research. They at least anticipate that other research teams will test the technology, develop their own enhancements, and create new use cases. When more trials are performed in this area, research group collaboration will probably rise. For instance, collaborations between Harvard University, Princeton University, University of California San Francisco, Massachusetts Institute of Technology, and Howard Hughes Medical Institute, among others, were highlighted in the Cell paper. The researchers claim that by working with several teams, they were able to better understand the workings of prime editing and improve some parts of the system. Also, the collaboration is a fantastic example of how thorough understanding can direct experimental planning. In addition to directly fixing defects, scientists are utilizing the technique to create healthy cells and organs for transplant. A change in emphasis from gene improvements like height, eye color, and body type to treatments and correction. Enhancing agricultural production and disease and pest resistance using prime editing. New crop varieties that are better adapted to various climatic conditions or growth circumstances might potentially be developed using this method. The development of novel bacterial strains and other organisms that are useful for industrial activities like the production of biofuels or the cleanup of environmental contamination. Increased employment chances for geneticists, biotechnology experts, and research laboratories. Thanks for watching. Request to subscribe and like. Comment your thoughts and views about gene modification.